Hello, everyone. I think we can begin today's meeting, and I hope that you can hear me well. Hope that the rest of the participants will join us as soon as possible. So today we have the third meeting of the series of seminars, Bioinformatics Club seminars, dedicated to the discussion of the role of artificial intelligence and state-of-the-art deep learning in bioinformatics. And again, just a quick reminder that the main goal of this series of seminars is to discuss what tasks can already be solved with all these state-of-the-art methods, large language models, and other nice tools. And also to think about what the future may be in terms of what the daily routine of a bioinformatician would look like in a couple of years from now, given all the pace of development of different tools and methods that can greatly aid us in data analysis and interpretation. And today we have two speakers who will talk about the role of AI and deep learning in analysis of proteomics data and in genomics and immunoinformatics. And I am happy to announce the first speaker. This is Dr. Yana Safonova, assistant professor of the Penn State University, and also a faculty member of the Department of Computer Sciences and Engineering at the Huck Institute of Life Sciences. We'll talk about the immunoinformatics part. So, Yana, please, the floor is yours. And yeah, then we'll proceed as usual. We'll proceed with the discussion. Again, encourage every participant to submit their question in the chat during the talk, and then we'll ask them afterwards.
Hi, everyone. Thank you for the introduction, Yura. I have some slides prepared, but I cannot share them.
Oh, yeah, that's my bad. Let me enable the screen sharing. Yeah, now it's enabled.
Okay, let's see. Do you see my slides?
Yeah, we did.
Okay, great. Let me remove all these floating things, and we can start. So I actually don't know much about machine learning, and in fact, I don't have expertise in this majorly, and I don't classify myself as machine learning specialist. I'm computer scientist by training, and I got my PhD in bioinformatics, and during my postdoc, I converted to what we call computational immunologist, but who really knows what it is. So generally what I do is computational biology. However, machine learning has such a dramatic development recently, and it would be just very silly of us to overlook this field just because we were not trained in it originally, because it offers so many opportunities, not just to do the research, but also organize scientific process in general. So what I'm going to do here today is to give you some of the overview of my field, which is majorly immunogenomics, so studying immune systems using genomic approaches, and also tell you about some of the directions that we start formulating where machine learning and artificial intelligence can be a very promising thing. So let's first start with talking about immune systems. Immunity is a part of our body that helps us to defend against various threats. Those can be foreign antigens such as viruses, bacteria, toxins. They can be our own inner problems such as abnormal cells, for example, cancer, and immunity can be related to immune-related disorders such as autoimmune disorders. In different ways, immunity is common for all living organisms, and invertebrate species is presented by two branches, innate immunity and adaptive immunity. Innate immunity is a very complex collection of cells and proteins that react immediately to an infection or an antigen. If you talk about a species or even a clade, then it's easy to compare innate immune systems because they operate in similar ways. We can easily identify genes that are responsible for certain things, compare them, and do all kind of statistical analysis that we usually do to see what's going on there. I'm not saying that it's a simple job, but overall we understand what we need to do. Adaptive immunity is a bit more complicated. It's actually a lot more complicated. It has only two components, B cells producing antibodies and T cells producing T cell receptors, but the diversity of these receptors is so big that we even haven't sampled it at all, even within humans. And because of this diversity, we still don't really understand how differences hidden there can be associated with susceptibility to diseases or create predisposition to certain conditions. And here is one and the major thing where we think eventually machine learning approaches will be useful. So before we start talking about them, let's dissect this diversity on a little bit more specific level. So we will be talking about this on example of antibodies. You heard a lot about them from COVID times, and antibodies are proteins that have this cartoonish white-shaped form, and with the top part, they can bind an antigen that can be a particle of bacteria, virus, or whatever. Once the binding happens, it can be recognized by other components of immune system, including innate immune components, and destroyed, or just antibodies bind virus and it becomes immobilized, so it can no longer infect host cells. So antibodies rely on the binding. And in order to get bindings to various antigens, we need a lot of different antibodies. And here is where the most complicated part is hidden. We cannot encode an infinite amount of antibodies just in case in the genome because genome is limited. So instead, antibodies relied on the process that's called somatic VDJ recombination that affects special places in the genome called immunoglobulin loci. This loci consists of several gene families, and VDJ recombination selects one gene from each family, concatenates them together, introduces small changes between the selected genes, and thus create a diversity of VDJ recombination that were generated from the same template genes that encoded in the germline genome. So VDJ recombination creates a lot of possibilities to recognize very different antigens. And once antibody encounters its antigen, it's also turned into an evolutionary tree where each node is a slightly mutated version of the original antibody. And the goal of this process is to improve binding through random mutations. It actually works like an evolutionary process with selection and mutagenesis, and the difference from the evolutionary process that's happening globally on Earth is that it happens very fast. Just within several days, we are able to generate good antibodies, while evolution that happens with living species takes place over billions of years, and we cannot observe it over a period of human life. So here you already see that there are so many somatic things that pretty difficult to study. And it actually was confirmed that they do result in very diverse sets of antibodies. If you compare antibodies of two different individuals, they will be having practically zero overlap. We do see exceptions from this rule. However, we generally do not rely on the fact that the same antibodies will be developed in a population. So here already we need to somehow represent the diverse set of antibodies into some set of features that are easier to compare when we talk about population-wide immune responses. So the first set of features that comes to mind is the original genes that are encoded in this loci. And here we are talking about 3 immunoglobulin loci. They are shown here with respect to the position in the human genome, and 4 T-cell receptor loci also positioned in the human genome. So what if we don't really look at highly diverse VDJ recombination and instead try to compare what is located in the genome? Would it be useful? It's actually proved to be useful in some applications. For example, there were individual genes that were identified to be associated with responses, for example, to flu or to specific COVID strains, or associated with development of autoimmune disorders. But overall, we still see that it's somehow fuzzy. And in many cases, we don't really have good associations. So could it be because the idea itself is not good? Or maybe because the genomics that is hidden here is much more complicated. And recently, we are getting confirmation that genomics is really tangled in these cases. So additional applications also include not just comparing responses across a population, but looking at very different species. Because here we are dealing with a system that has so little constraints and so much opportunities to be diverse, we sometimes can look at unusual species to see if they have unique immune adaptations. Examples can be quite interesting. If you look at cattle, we see that they have antibodies that can neutralize HIV. If you look at frogs, then their skin contains antibodies that can very efficiently neutralize microbes, so they can be as a supplement to antibiotics. Snakes are believed to neutralize their own toxins, so it doesn't poison them. Birds somehow tolerate many viruses, including flu. So the list goes on and on. And if we learn how to dissect this diversity and analyze it, we might be touching a base for development for very diverse drugs that were already shaped by evolution. However, it's again not a simple problem, because to do this, we need to go to the genome of every species and do accurate annotation. As I said, population analysis, aiming to find associations and explain why some individuals respond better or worse to specific diseases, is a huge application, especially in human health. However, it also can be applicable for agricultural animals, so animals that are important for our well-being, animals that live close to us and occasionally can transmit diseases, but the patterns are tangled. So again, the list is very long. And finally, this analysis is important just to understand how it all works together. It's called the One Health approach, when we don't just look at one single species, but we look at them all together, and we know that the species have different roles in the ecosystem. Some of them can be reservoirs for diseases. Some of them can be amplifying hosts. Some of them can be model organisms. But because adaptive immunity is so diverse, how exactly do we do this comparison? So immunogenomics is aiming to answer this question as well. And here is additional piece of results showing that this translation is quite challenging. So let's look at the human and mouse. Mouse is a standard model organism, and if you just collect the immunoglobulin genes, ones that encode antibody responses, and create a phylogenetic tree, we will see that there are no orthologous gene pairs. we see some ancient orthologous groups located in the same subtrees, but overall there are no pairs that would provide us with a basis for this comparison. So for example, we learned how disease works in mouse, and we know what genes mount successful responses. So what's next? How can we say which genes would do the same in human? Looking at this tree tells me that it's going to be very complicated. If you look at the macaque, which is another popular model organism, we see that even if we narrow it down to the primate clade, it still would be quite complicated. There are many species-specific expansions that often contain highly expressed genes that again would make this analysis very fuzzy. So we only see pretty good correspondence if we look at the level of human and bonobo, because they are two closest species if we talk about human, but no one is doing experiments on bonobo because they are endangered, it's highly unethical, and overall we just need to solve the problem on the top or in the middle. Recently, we did an analysis that showed us that not just genes are extremely diverse, but the whole genomic regions that encode them are very unique to a species. And with our students at Penn State and Cornell universities, where my collaborators are, we showed that they mutate very fast. So on top of somatic mutations that we see in antibodies, we also see mutations in the germline genome. And this mutation rate is extremely high, and it's not single point mutations, though extensive structural variations that make everything very different, very complicated. So here is another connection with artificial intelligence, because we might need to find this pattern, the patterns in the structural variations to understand what's actually happening with immunoglobulin loci to eventually create a bridge toward translational studies. However, I also wanted to show you what's going on at the population level, because it might be a simple case to study, but it also shows us the beauty of the system and how rapidly it evolves and how different it looks from other genomic regions. So here is a representation of structural variation in the human immunoglobulin heavy chain locus, one of 3 immunoglobulin loci. This was our study on 150 individuals that we sequenced with colleagues. And overall, there are many mosaic variations, deletions, complex things, but I want you to look at this particular region. Before we do, let's look at the standard representation of genomic architecture called dot plot. It's actually a very simple plot that shows all local alignment between 2 sequences. If you align sequence against itself, it shows that the main diagonal, so each dot here, point here can be projected to X and Y, and obviously identical positions correspond to aligned positions in the perfect alignment of sequence versus itself. If you have repeats, they will be also detected and will be shown as additional diagonals. Again, we can project each position to the X and Y, and they will correspond to positions of corresponding repeats. If we have inversions, we will have diagonals looking at the different direction. And overall, this is a very simple way to very quickly look what's going on with the sequence. So if we look at this region of interest, we will see that it's actually a tandem repeat of multiplicity 1, 2, 3, 4. And here it makes sense that it's filled with structural variations and specifically deletions across people, because people might have different compositions of repeats here. And if you look at our close relatives, remember, we looked at human versus bonobo and saw that there are a lot of correspondences. So maybe that is unachievable, but still ideal model. we will see that our closest relatives, such as gorilla, bonobo, and orangutans, also have expansions at the corresponding regions. Sometimes these expansions can be contracted, sometimes they can be overexpanded. Specifically in bonobos, they expanded. In the gorilla subjects that we selected, they were contracted. And this also shows us that these regions might be actually the single driving points of this rapid evolution that we just observed on a high level when we compared many different species and found out that the immunoglobulin loci are extremely diverse. So if we decompose the tandem repeat into individual units, which is not complicated when the structure is simple, we can compute the phylogenetic tree. And then we'll see that expansions that were observed in bonobo and human are actually species specific. So it means that they emerged independently in these branches, and they do not have perfect correspondence with each other. So here is actually a very important thing that showed us that we can pinpoint this small event that eventually results in very different loci. And it also tells us a little bit on how to create biologically informed pangenome. Pangenome is a buzzword right now. Everyone is doing pangenomes. But when it comes to complicated regions like this, and often these regions are biomedically relevant, we sometimes make pretty bad decisions. So here is an annotation of the tandem region for these four species. We can do maximum parsimony, and in this case, it will be looking like this. However, that's not what this phylogenetic tree is telling us, because if we clump human copies that are specific to humans and bonobo copies that are specific to bonobos, we will basically ignore information that the phylogeny is telling us. So the actual biologically informed pangenome should look like this. Even if these units are similar to each other, we shouldn't clump them together just because they can be aligned. We actually need to learn from the evolutionary history and see what's going on there. We eventually saw that if we add more gorilla haplotypes, we will also see gorilla-specific units that are not shown here, saying that we also here hitting a selection bias if we making an assumptions about a population while looking at the limited number of genomes. So this example was quite simple to analyze manually, but in reality, when we start looking at the whole collection of structural variations there, it's getting more and more complicated. Even here, even on this species, we have another regions where diagonals break, where this analysis is getting more complicated. And actually learning from these plots might be another cool application of machine learning, because it can capture some of the patterns that are difficult for human eye to detect. And to wrap it up, this is, of course, very interesting from the fundamental point of view. I'm completely fascinated by adaptive immune loci, how rapidly they evolve. We see that sometimes two species are very similar to each other, and when it comes to immunoglobulin loci, they just have very different sequences. They cannot be aligned. Like, why on earth this is happening? This is a very cool question on its own, but in the end of the day, we are interested in practical applications. And practical applications tell us to compare immune responses, we need a comparison basis. We need either to put genes that are similar to each other, that can break down the adaptive immune response signal to see some associations, or we need to somehow represent this comparison basis from antibody repertoires and these highly diverse genes that are used for generation. So this is actually a question for statistical methods and machine learning methods. It can be very complicated if we start looking at domesticated animals, for humans, it's complicated, but for cattle, it's extremely complicated because basically cattle loci are whole formed by tandem repeat, and it can expand very uncontrollably. So again, if you have two cows, how do you find correspondence between their genes? If you have cows and if you want to see how they transmit their diseases to humans, how do you find correspondence between this and human? This is a question that I believe machine learning would eventually need to solve. And here is directions that I highlighted in bold. Basically, learning as much as we can from antibodies, immunoglobulin loci, and creating better representation as a collection of features that can be uniformly computed across and within populations and compared, and also trying to find hidden patterns there that can be represented as a complicated combination of those features. And two more directions that are not directly related to my research, but they still fall in the areas of antibody and immunoinformatics is actually determining specificity of antibodies and computing is three-dimensional structure just by looking at the sequence. So yeah, the latter can be done with AlphaFold for other proteins, but antibodies are still exceptions from this rule, and current developments do not offer the same level of accuracy as it does with other proteins. And because antibodies have this robust system to change, to accumulate mutations, to have better binding, that can be another cool application. Like if you have some starting antibody, can you apply machine learning from that was trained on existing databases that would introduce mutations that make this antibody drug better for binding or from thermostability point of view. There are studies that already was doing this, and I'm pretty sure that among these four directions, the last one will be one that is implemented much faster compared to others. So here is my completely biased overview. There are a lot of people involved in this research. Thank you for listening. I'd be happy to take questions.
Okay, thank you very much for a nice talk. As usual, to make the discussion a little bit smoother, I ask and encourage people to submit the questions in the chat. And if you don't mind, I will begin with a short question regarding AlphaFold, because again, this is probably one of the most significant achievements of these state-of-the-art deep learning models in the recent years. Why is it not capable of properly folding antibodies? Do you have an explanation as to why they are such a hard target for AlphaFold?
Because they have different composition of amino acids that is not seen in standard proteins, because they are shaped by binding with antigens. And first of all, because the database that we have right now is extremely biased. It's mainly formed by either flu-specific antibodies or HIV-specific antibodies. And we can pick a lot of good things from there, but it won't be a versatile solution. And historically, this database is limited because of the cost. It's expensive to generate three-dimensional structure, and the number of existing antibodies is still quite limited, I would say several thousands, but to cover a lot of possibilities, we need more.
Okay, thank you. And maybe also one question that I always ask to every speaker at these seminars. So again, we have all the recent sort of buzz around the large language models and their applications. And Most recently, I think a lot of the attention has switched to developing sorts of so-called agentic workflows when you have like automated data analysis, sort of prompt-based bioinformatics. And if you could comment on where do you see the like the most promising area of application of these particular types of models that can handle primarily text as inputs? What do you see their scope in this field, if any? And what do you think about this like prompt-based bioinformatics software?
Yeah, overall, I think coding in programming in general and actually scientific writing will be the first thing to be transformed by AI because many routine things that we do manually is now much easier to do it using AI. I see no harm in it. It will definitely change the way we teach those kind of things because like just recently, I created a curriculum for course on algorithms and data structures, and we have a special option to increase your final grade by providing solutions to code force problems. Well, with ChatGPT, it's no longer a challenge, so we probably need to come up with something else. But in general, I use it uh, very often. And again, if I have like a collection of scripts that I need to put together, I just say, hey, ChatGPT, could you please create a pipeline that takes this scripts into something much faster and better? And yeah, that's actually a great application. The same with scientific writing. There are so many things that we write over and over, especially in technical texts like grant applications and some maybe parts of the paper. Again, I see no harm in it as long as it reflects your final curation and your original thoughts. Because if we need to reflect how, for example, some scientific direction influences the society and other important parts of humanity, well, yeah, I would like for this part of the text be generated with my original bullet point. So again, it's easy to abuse it. It's silly to ignore it. We need to find balance, and I don't think that currently I'm in a position to answer where this balance is.
Okay, thank you. Yeah, one more question that was in the chat from the very beginning of your talk from Paul Sanitsin. The question is, why is the VGJ recombination called a gene recombination instead of exon recombination?
Because they are not exons. Exons undergo splicing, so here they actually it's just VDJ recombination, but we call the things genes, even though they do not perform as genes in the original sense. Sometimes people call them gene segments. I think that's one of the examples where it kind of breaks the standard notation. So we call them genes because it's shorter, but I would say that it's neither.
Okay, yeah, yeah, let me then follow up. I see that there's one more question in the chat, so just a moment, I will read it up. But before that, maybe you could also comment on from the biological point of view. What's probably your explanation for the differences that you see in the specificities, antigen specificities for antibodies from different species? So why are these trees that you've shown so different? Why do they show this clustering?
Yeah, this is a fantastic question. And again, we recently published a study when we showed that one of the factors that can shape the architecture of immune loci are genetic bottlenecks. We found that when the population is reduced, mobile elements starts to jump more actively, probably because the suppressing mechanism do not work that well. And species that actually went through bottleneck at some point in the past start looking more repetitive, and eventually they either converged into this repetitive pattern, for example, as cattle and all domesticated animals went through this bottleneck artificially. But some species eventually recover from this self-repetition, they accumulate mutation, and they just create different layout. So I think it's just one lens on how to look at the process. So like imagine, even if we started from similar sequences, but we went through different trajectories and we hit population bottlenecks, we repeated different parts of the genome, so they are already no longer identical, and then they mutated, and then we may enter population bottlenecks as well. So that's what we found so far, but I think there are other genetic elements that can be associated with increased flexibility and fragility of these places, as they are just also designed to be fragile, to be able to change fast and to generate new genes faster. I see also a question in the chat, and I think it's related to what I'm currently telling. For example, we see sometimes that species come up with different ways to create immunoglobulin genes that can mimic other host proteins that can be bound by antigens. So when antibody mimics the host protein, it also can efficiently bind the antigen. And it's very surprising, but from this highly diverse loci, we still see that these signatures of molecular mimicry of host proteins can be developed independently. And that's, I think, is one of the powerful angles and examples how we can integrate advanced statistical methods to actually create some interpretation of this diversity, not just tell that, wow, it's so different. So our current model that we are looking is CCR5 mimicry. CCR5 is used by various pathogens, and we are particularly interested in some of the applications. For example, there is a cool application for endangered ferrets in the United States and Canada that are particularly susceptible to plague, and we believe that CCR5 plays a role there. And on the other hand, it was shown that CCR5 mimicking antibodies mount neutralizing responses to HIV. So wouldn't it be cool to finally develop something HIV related? And you already see like plague, HIV, completely different antigens, very different pathogenesis, but maybe the link connecting them is something in common. We also started looking at reservoir species like black-footed, those are ferrets, white-footed mouse that are reservoirs for Lyme disease. We look at agricultural animals a lot because they interact with each other. They interact with wild on the one side, with human on the other side, and they kind of are mediators to transmit diseases like brucellosis, tuberculosis, sometimes some prion diseases. I'm not sure that it's related to immunity, but again, they are in priority because of that. And here we're just trying to understand what exactly all this domestication means. So that would be another example. And recently I started working more with conservation scientists who work with endangered species, and in every endangered species, there is also a disease component. So the question is here is again how much we can get from genomic studies to understand the One Health approach.
Thank you. Yeah, if you don't mind, I'll ask one other question from the chat just to get more questions in. So There's a question regarding what is the main factor contributing to the preventing the large-scale application of immunogenomics. Is it the availability of data on some associations, or is it just a question of modeling? So what's your take on the main limits of?
Yeah, I think we still don't know much about the biology of it. So from the beginning of immunogenomics, people started to implement some machine learning approaches to see some commonalities. And the results were always not as good as we wanted them to be. So first of all, because there were unstandardized protocols for sequencing. People created sampling bias and sequencing different parts of antibodies. When we actually looked at the same sample sequenced in two our collaborators and found zero overlap, I was like, what the hell we are even doing? Like, we can find all kind of associations, but they will be wrong. So eventually people started to apply the same protocols. But next, we started looking at different people, and we learned that they have so much germline diversity in them. So it's like, I have this gene, but you might have two copies of this gene, and you might have a gene-converted version of this gene. So again, what is the basis for comparison here? So we started doing pangenomes. And once we learned from pangenomes and maybe started doing this biologically informed pangenomes, again, it's a concept, not exactly a know-how. Maybe there is going to be another layer of complexity, like, I don't know, 3D structure. So overall, I would say it's very cool what machine learning and AI is doing right now, but let's not forget where we're coming from. We still need to understand the fundamental part of it. We just need to treat it as a tool, not as a crystal ball that will give us all the answers, because the answers that it will give us will be trained on our current understanding. And from what I see from genomic studies, we don't understand much. But isn't it exciting? We will have jobs a little bit longer. So, yeah, I think it's work for all of us.
Thank you very much. Yeah, that's a really good point for us to end this first part, because yeah, that's a question that I also frequently asked during these sessions is whether we can expect all to be replaced eventually. Yeah, let's hope that
not that
that will not
fast,
happen.
I think.
Yeah.
But it's our
Okay.
job to translate these results in the way that they people understand them. For example, finding agencies.
Perfect. Yeah, I suggest that we will move on to the second speaker because we are running a little bit tight on schedule. And I would ask Yana, please, if you can reply in the chat to the remaining couple of questions that are there. That would be great. Yeah, thank you again for your presentation. And with that, I'm happy to announce the second speaker, Dr. Paul Sanitsen from the University of Utrecht from the Netherlands, who will talk about AI in proteomics. Paul, please, the floor is yours for your talk.
Yeah, sorry, I just finishing the question to Yana. Jan, thank you a lot for presentation. That's actually very nice. And indeed, there's too many things unknown in antibody world. And I also a bit lucky enough to work a bit in this field, but from a slightly another side, not from evolution side. And that's a very exciting topic indeed. Okay, great.
Thank you, Pasha.
Yeah, so you can see my screen, right?
Yeah, we can.
You can see my slides. Yeah, one thing would be really cool if people will switch on camera. I feel like would be a bit nicer. Meanwhile, so today I'm going to talk about MS-based proteomics and more about like how machine learning and AI. But I'm a bit modest person. I don't like to use AI in every sentence. So I would say how machine learning advancing our topic, our field. So yeah, just a bit about myself. Indeed, today I'm working as assistant professor at Utrecht University, but before I was doing my postdoc, was doing my bachelor and master thesis in Lomonosov, Moscow State University. I was doing my PhD in Max Planck Institute, doing actually proteomics. And my postdoctoral research also was focused on proteomics. And yeah, no surprise, my lab is also focused on proteomics. So what's the agenda for today? So we're going to discuss why proteomics and why using mass spectrometry, because indeed there's alternatives, and it's actually very important to discuss them. How to do mass spectrometry-based proteomics. And next, I decided to slightly kind of separate two topics: machine learning AI for MS method development and machine learning AI for mass spectrometry applications. So, and in both of these 3 and 4, I'm going to discuss what's the current state and what would be the future. And yeah, and feel free to write questions to the chat. Okay, great. So why proteomics? Because proteins, as we also seen from previous presentation, we do have a lot of fun, a lot of life. So we can be, we do have a turnover, we do have a half-life, we can be post-translationally modified, we can be localized in different places. You know, there's alternative splicing, there's a lot of things happening with them. And that would be really fun to know what's actually happening in our sample. And that can be whatever. It can be blood, it can be saliva, it can be spinal liquids, it can be cancer tissue. So basically, if you look at each level of sample dogma and which kind of degrees of freedom do we have on that level. So like, let's say DNA, we have combinatoriation, mutations, methylation, RNA expression splicing, but proteins basically integrate all these degrees of freedom. And definitely that would be important to study, all right? I hope I did convince you enough, but so, and now I'm coming to the methods. So one of the methods of actually studying proteins, but also metabolites, lipids, and that's kind of basically a very uniform method for studying all these biological molecules. That's not just biological molecules, actually everything. It's mass spectrometry. So in simple terms, mass spectrometry, it's a device, usually like indeed either magnetic, either electrostatical, physical device, which basically just constructing such spectra for molecules. So on one axis, I'm sorry, I didn't write the axis. Y-axis is intensity, and X-axis is mass to charge. And you should trust me. That's a single peptide with such sequence is present here. Yeah, and that peptide had been measured with different resolution. So it's a bit strange metrics, but that's not really important. So 1,000 is very low resolution, 100,000 is something we do every day, and 1 million it's crazy resolution, very for special purposes. And trust me, this is one peptide with charge free. But can somebody tell me why do we see not just one peak with one mass? Why do we see several peaks with several masses? Maybe someone saw that plot before. So the truth is that kind of if you calculate how many carbons in this peptide, the number is going to be over 100. And maybe you know one of the most frequent isotope in the world, naturally abundant isotope, it's a carbon-13. So the most stable, the most abundant and stable, it's C12. But there's also C13, and that's actually a significant portion. That's 1%. And if you imagine if there's a molecule with more than 100 carbons, there's a really high chance that you detect at least one, molecule with at least one carbon-13. And that's basically what is driving this several peaks. So basically, that's a peptide with no carbon-13 and with specific mass divided by charge. But now if you add +1 neutron, which comes from carbon-13, you kind of you basically see another molecule which mass +1 divided by charge, etc., etc., etc. So you also can find out the distance between these peaks going to be 1 divided by charge, which is very helpful. So yeah, that's a very important thing to keep in mind. We indeed, for each molecule, we see not just one peak, but we see multiple peaks. And that's going to be a way more difficult, more complicated task if you will study whole antibody, let's say. That's a whole huge protein, not just with 15, 20 amino acids, but 150 or even more. And that's isotopic distribution going to be very biased. It's going to be biased towards, you will not see monoisotopic peak whatsoever. Why do we study why do we use mass spectrometry in the first place for studying proteomics? Because we do know that amino acids are modified a lot, and we call it like there's an umbrella term called post and translational modifications. But if you know something about modifications in RNA or DNA, that's basically outshadowed by so many different modifications in the case of proteomics, because amino acids are way more diverse in the sense. And many of these modifications have very important signaling properties, etc. So one of the simplest example is probably phosphorylation, but also there's a glycosylation. There's tons of different modifications. Yes, and we do indeed can detect these modifications thanks to the tandem mass spectrometry when we actually fragment these peptides and we see mass shift of one set of the suffixes or one set of the prefixes. Anyway, so one thing I have to tell you that there's multiple flavors of proteomics, mass spectrometry-based proteomics, and one flavor is called shotgun, and another one is top-down. If you heard shotgun term in terms of genomics, then that's fully translates to the proteomics. So for us, we're saying it's hard to study proteins, intact proteins. Indeed, it's hard. Trust me, please, it's a really hard task. That's why we digest these proteins on the peptides. And so basically now we're studying all these peptides as a separate instances. And in order to sequence each peptide, we fragment them in the gas phase and study all these fragments, and that allows us to identify them. So that's a shotgun proteomics. And indeed, if your task is to detect, let's say, 10,000 proteins in your sample and quantify all of them, you rather do shotgun proteomics. Top-down proteomics is still really hard to do such things. I would say impossible for now. Yeah, so that's basically summarize what I described before. So we can take any tissue, we can take cell line, we can take blood. We extract proteins. We don't like to study intact proteins. We like to digest these proteins. We're getting multiple peptides. Imagine it's going to be, let's say, 15 genes. Out of 15 genes, we're going to get something like tens millions of peptides. It's really, if we throw all of these peptides at once, we're going to get very complicated spectra. And that's why always peptides being separated with HPLC. So basically with liquid chromatography. And all these peptides are living in a liquid phase, by being transferred to the gas phase, simultaneously charged with electrospray. And this was a huge breakthrough. That's this transfer from liquid phase to the gas phase, and charging was done softly. That's actually Nobel Prize for chemistry in 2002. So, and now we have a mass spectrometry mass analyzer instrument, which is building spectra, measuring spectra, let's say, spending 20 milliseconds of each second to measure spectra of intact peptides. And we do see that some peptides elute with hopefully normal distribution. Some peptides are finishing elution. You see the blue one completely off, but the orange just appearing. And that's how it looks like. So basically, it's a bit of Alps, some peptides appearing, some peptides disappearing, but at least we simplify our life, not measuring all peptides at once. And that's very important. So, but at the same time, okay, cool. We see some peptide, we see the isotopic pattern, but the question is, which peptide, which sequence, what sequence of this peptide actually is? So, and in order to find the sequence of the peptide, people come up with the idea of isolating this peptide in a gas phase. That's actually doable. This peptide being isolated and fragmented. So the idea of fragmentation, there's a multiple way to fragment peptides in a gas phase. One of the simplest one is just to collide it with a neutral gas and send this kinetic energy into breaking the backbone of the peptides. And that's what we get. So basically, hopefully, you get all possible prefixes because of the prefixes and suffixes. So basically, prefix which contains the first amino acids, and suffix contains the last amino acids all the time. And hopefully you'll find out all possible fragments, which is unfortunately not the truth. Anyway, okay, cool. Then but the reality is that although I show you that the spectra have been generated by peptide called peptide, so basically each character here is amino acid. And you know, in reality, we're getting some spectra, and who knows which peptide did that. And that's actually quite separates mass spectrometry from NGS in terms of that in case of NGS, you're getting sequences from machine, which don't need so much interpretation in terms of what kind of sequence Illumina generated here, right? So, but in the case of mass spectrometry, indeed, there's a step of interpretation. You're getting some spectra, and now you're asking, okay, which peptide could make that? And there's a kind of two approach how people dealing with this task. The first approach called de novo assembly. So basically, we know masses of amino acids. We do believe that we do find most of the prefixes, most of the suffixes, and we also know there's distance between these fragments going to be masses of the amino acids which have been fragmented out. So, and that's kind of basically we see do peaks, and this between them the mass is definitely T. Then we're trying to assemble all these peaks together, hoping that that's going to be representing some specific peptide. Or as you see in this case, PT, we miss some peptide fragment here, and that's why we don't know order of this. It can be PT or TP, who knows? And the idea is just from the spectra, just based on the masses of the amino acids, we'll be able to find out that's such peptide generated that, and that's called de novo assembly. Or another method is saying that we know that we're analyzing human sample. Then we know the reference FASTA file for the proteome of this sample. So let's basically just generate theoretical spectra of all possible peptides, knowing the protease which we used. And let's try to match this observed spectra to the theoretical spectra. And that's kind of matching against database is all about. And hopefully the best score identification would be exactly the score which correct identification of a correct interpretation of that spectra. So that's basically it. So there's two methods, matching, like database matching methods and de novo assembly. And I would say if you're from NGS field, the best analogy would be that if you're analyzing some transcriptome of unknown species and you don't have reference genome, then you probably will go for de novo assembly. It's going to be computationally expensive, but yeah, at least you will assemble you will assemble some transcripts. But if you study human proteome, yes, indeed, you can do de novo assembly. But you know, human genome has really nice reference genome. So then instead of basically assembling it, you can just align it on the human genome and move on. So yeah. And the topic of today is, one comment I have to say. Machine learning, mass spectrometry is not a single method to study proteins. And if you heard something about O-link or SomaLogic, so basically it's a method of having some antibody-based or DNA-based adapters against proteins, and measuring quickly these adapters is basically a method of really scalable way of studying behavior of concentration of proteins across thousands of samples. But again, if you come to the question of, okay, what's about post-translational modifications, or intact proteins, probably mass spectrometry is still method of choice. And yeah, now we come back to the topic. What's about machine learning AI, and how this method helps for mass spectrometry method development? So I would say currently I would highlight 3 topics: target decoy separation, gas liquid chromatography prediction, intensity of fragment spectra. So I would say in terms of complexity, the last task is the most complex. But yeah, so I decided to describe target decoy separation first. So the thing is that when this matching happened, we come up with some quality metrics called score. And the thing is that in order to control false discovery rate, we're actually adding to our database not just reference a digested proteome, but also decoy digested reference proteome. And usually it's actually just reversed peptides, just like that. So why we adding decoy in the first place? We adding decoy just to control false discovery rate. So we're basically saying that whatever map on decoy database is wrong, for sure. Whatever will map on target, it can be true positive, and it can be false positives. So basically, in order to quantify what's the false positive ratio of there, we try to use the decoy database kind of to simulate, to emulate how the wrong portion of the target identification looks like. But the problem is that if you plot this target decoy hits, and target's going to be blue and decoy going to be red for different lengths of the peptide, so basically 6, 8, 10, 12, and basically until 24, you will quickly find out that so target and decoy are almost unseparable for the length of 6. And there's some separation happening on length of 8, 10, etc. But basically, if you decide to come up with some smart way to make cutoffs, then you will need to make a cutoff which is aware about the length of the peptide, correct? So basically, you see that's probably the best cutoff here would be here, best cutoff for 12 going to be somewhere here, and probably you have to drop the idea to make any cutoff for the length of 6. And that's in the past was called the posterior error probability. I told you about length, but it's also a sequence or charge also take into account. Basically, if you know distribution over all decoys over the sequence and length, so you basically can modulate and measure what actually we need to know. We need to know, take into account the sequence and length. What's the chance that this identification was wrong? And that's basically was beginning of everything in terms of statistically robust identification. But I must say, slowly we come up that machine learning and deep learning can do a really good job. So it's becoming especially useful when amount of the features, not just length, but it can be way more. Let's say right now we're dealing with identifications which have 20 features, and machine learning doing exceptionally good job. But I must say, you have to be cautious. You should not be all the time over optimistic. I would like to, for that problem, I will go ahead and use billions of parameters. The deepest machine learning I will do, or even AI, I will go ahead and do it. I would rather all the time I share the idea that let's start something modest with something super simple. Let's start with simple SVM, and slowly kind of increase the complexity and find out when complexity is already there's no need for more complexity. So, and that's kind of the idea here. It's very much shared by field, I guess so. So the second task that's quite simple. Let's predict some liquid chromatography predictions. You remember I told you that this peptide leaving a column with specific time, and can we predict that? That's actually a very simple task. Apparently, we can predict retention time of the peptides with super high accuracy, and that's indeed true. Yet another chromatography exists that's called ion fractionation. So it is collision cross-section, also easily predictable with machine learning methods. But also right now there's a quite shift towards using protein language models as basically default feature extractors. And on top of these feature extractors, people are building a fully connected neural net. But of course, it's easy to build. But another step is interpretation of all these things, which is, of course, have to be done in our field if you want to convince anybody to use these prediction tools. Yeah, but as I said, the most difficult part is intensity of fragment spectra. You remember I showed you this plot, right? So, but you see a fragment prefix P and prefix PE and prefix PEP and PPT, PEPT, and all of them have very different intensities. And you can even like find out PEPT and EDE. So basically this complementary, because this is prefix pept, and ide is the suffix which complements until the end of the sequence. So basically, even that suffix and prefix have different intensities. The question is, can you predict which fragments going to have such intensity, etc.? The truth is that with physical chemical properties, with physical modulation, it's actually hard to do. And apparently machine learning doing actually a very good job on predicting these intensities. At least order of these fragments in terms of intensity doing a really exceptionally good job. And that's basically the whole story of Buzz thing happened 2019, but of course that was many papers before. And basically if you take sequence of the peptide as it is, take charge of the peptide using some deep neural net or neural net, can we predict actually intensities of those fragments? And the truth is, yeah, we can. And people use many different methods. I think it's still quite a topic which people trying different methods. I feel like at the beginning, many of us, including me, we were trying bidirectional LSTMs, different recurrent neural nets, and we did a good job, but there's one issue with them. That's we are slowly learning. So, and right now, probably encoders is one of the best approach to compress and transform, and encode sequences into some meaningful space. So yeah, that's still happening. But one thing I would like to highlight, it's used a lot. Right now, it's many labs are indeed using predicted spectra as a state of art. So as a kind of normal part of the pipeline. So that's pretty normal now. Yeah. If we talk about ML AI for MS methods developments, so what's the challenges? I'm not going to go through each of them, but I'm just would like to talk just briefly. So what's kind of, from my point of view, people as a community, we're all working with that, but what is still kind of could be improved from my point of view. And that's kind of field of active research, let's put it this way. It doesn't mean that previous topics are not active research, but I feel like here I would call a bit of like hot topics, currently hot topics. So I would like to highlight the 3 topics that's predicting digestion, solubility, and flyability biases. So basically, very simply, we're studying these proteins with shotgun proteomics. We need to digest these proteins on the peptides. And although all these peptides, which highlighted with 3 colors, coming from the same protein, let's say, even forget about splicing, all these peptides coming from the same protein, we end up with different intensities. And that's ridiculous. So basically, we do have multiple peptides and all of them coming with slightly different densities. And the question is why? Because there's a multiple biases. And one of the probably clear bias is a protease bias, because this enzyme which has some preferences. And let's say if we write digestion probability on top of each amino acid, that's not going to be a 1-0 split, unfortunately. There's another bias called solubility, So if protein is if protein loves water, hydrophilic, if the protein hydrophilic, it doesn't mean that the peptide, any random peptide from this protein is hydrophilic. And that's actually a very mind-blowing thing. Some of them can be exceptionally hydrophobic. And as a result, once the protease digests this peptide can be non-soluble in the liquid and just drop to the bottom of the Eppendorf. That's another source of huge variability. And another source is basically, imagine perfect situation. All of these peptides are hydrophilic, and all of them have exactly the same intensities, and you pass it through the through electrospray, which is basically transferring from the liquid phase to the gas phase and charging. Believe me, all these peptides will not result with exactly the same intensities, unfortunately. The question is, can we use machine learning for that? And obviously people trying, but in the same time, it's not as simple as it could look like. It's not just, let's predict bias for all these 3 steps. The truth is that all these protease biases, it's kind of, we can detect this peptide between P and K. So there's a kind of inclusion, exclusion. So this data are quite complicated in terms of how can we modulate, how can we model behavior of all this process. And indeed, just stupid dummy usage of machine learning, I think it's not enough, not anymore, let's say even for publication. So yeah, Another topic is end-to-end de novo spectral interpretation. So when people taking just the raw data without any interpretation of the data, no filtering, no denosing, let's say, we're just taking a set of the masses and intensities and feed it to the transformer using like peak encoder first, position encoder, and basically using transformer decoder, we're trying to take this MS spectrum and trying to predict next amino acids. Let's say we found out already that there's a peptide, but are we able to find out E amino acids as the next amino acids? That's a good question. But again, what's I think the danger here that we have to bring machine learning which is capable of dealing with uncertainties. So uncertainties that sometimes machine learning should say, I don't know. So and that's actually a bit problematic, that's, for example, my lab currently trying to work towards this direction, add a bit of uncertainties, especially when we're trying to sequence antibodies. Actually, so indeed, we're trying to sequence antibodies which are big proteins, and asking machine learning to be uncertain at specific places. That's okay. That's okay to have mistakes. So anyway, and last topic, which kind of machine learning for method development, I would say it's a spectral foundation models. It's indeed kind of people trying to improvise with the idea. If we take this, let's say LLM, how LLM trained, we take the one word out and ask machine learning to predict on the contextual information around which word we removed. That's the same idea here. So let's say remove peak and ask machine learning, can you predict intensity of this peak which we just removed with knowing mass? Or if we know intensity, can you predict the mass of the peak which we removed? Or can you predict both of them? And I think like that's quite a super hot topic right now. Many people trying to do that. So people published super nice paper recently for metabolites. That's a highlight of this research. I think I hope that will help us to interpret spectrum better without introducing any pre-processing biases. But let's see. Yeah, I know I'm running out of time, but I quickly show maybe a few more things about applications. So I kind of highlighted here that's overall improvement of identification. Indeed, machine learning helping a lot, and indeed helping a lot, especially for DAA proteomics. I did talk a lot about DAA proteomics, but let's say if you want consistency, proteomics over 1,000 samples, that's method of choice. So, and machine learning help is real here. Another thing which I personally like a lot, improving of immunopeptides representation predictions. I feel like it will have a lot of practical consequences, and machine learning and mass spectrometry in general helping a lot there. Another thing which I just wrote, advanced differential expression analysis, binary classification. I would say people more and more using kind of XGBoost or a bit more advanced statistical methods to actually classify thousands of samples and find out features which are more important than another, etc. I think it's also worth of highlighting that is becoming more common practice. But one thing I would like to say, it's immunopeptides representation. So basically we know, yeah, it's slightly another side of immune story, which Yana told us today, but basically, maybe you know, every cell has 6 MHC proteins, HLA1 proteins, 3 genes, so 6 alleles, and all of them are different. And that's probably a good thing. Basically, and these 6 alleles are very different from me and probably also Yana and from any random person who is listening to this presentation. Honestly, if you take all these peptides from the surface from which loaded on these MHC proteins, that's going to be a very complicated sample. And it would be hard for you to understand from which out of 6 MHC this peptide came. So what people did, we engineered cell line, we removed all the 6 MHC complexes, and we start inserting one by one. I have to say, each of us have 3 genes of MHC, but these genes are highly variable. It's one of the most variable gene in the human population. And basically, we just took top 100 most popular HLA alleles and inserted one by one into the cell line. And basically now we know which HLA protein we have, and we see which peptide are eluted from such a cell line, and using mass spectrometry. And basically having this couple information, that was a kind of gold mine for improving basically predictions. If these somatic mutations will be present on the surface, if these somatic mutations have some potential for being detected with immune system. The short story, I hope in something like 10 years, we're going to talk about personal vaccination against cancer. And that's kind of very much real from my point of view. So we will make exome sequencing of cancer and blood, and we will find out which somatic mutations have a chance to be present using improved models with machine learning and mass spectrometry. And yeah, so that's what's going to be used for vaccination. So, okay, and challenges. From my point of view, there's a very, okay, also all challenges, all current development which I'm writing, that is purely my opinion. I'm not covering everything. And that's almost last slide. I feel like challenges which are super exciting, it's a protein-protein interaction predictions. Mass spectrometry has techniques to predict which proteins interact with this specific protein. And we see many of them. And I still believe it's not used a lot by community to predict protein-protein interactions. Indeed, you have to take structural information into account, carefully deal with this mass spectrometry data. But anyway, I feel it's actually a huge challenge, but it will be used, I'm sure. Another topic is PTM-aware structure predictions. That's kind of, we didn't work with this directly, but I would like to show you a few slides which will inspire you for this work. And of course, the challenge, grand challenge, which I love to participate right now, it's antibody repertoire sequencing. So basically, if we take blood of the blood droplets, can we sequence all antibodies which are present in this blood, or can we see how antibody repertoire will change after bone marrow transfer, after chemotherapy? This is freaking awesome questions, and indeed one of the biggest questions, which scarily we don't know properly, kind of how antibody repertoire changed after vaccination, for example. So like across population. That's super interesting questions, but we need to sequence these antibodies, and that's what's actually very active work in our group is focused on. But today we're going to talk about PTM-aware structure predictions. I know, but I will be very fast. So basically, in one paper, which is shown here, published 2024, we basically took mice, took many different, multiple tissues from these mice, and we did phosphor enrichment analysis. So basically, we found, I don't remember, tons of, let's say, tons of phosphor sites, phosphor-related peptides. And if you kind of do some dimensional reduction based on the all binary comparisons of all these peptides, I think I even downscaled it, but it was really something like 100,000 or 80,000. So you find out that indeed there's one huge cluster with SP. So basically, it's indeed a very famous pattern for phosphorylation. This serine threonine really love to be phosphorylated if it stands before proline. But as you see, it's not a single pattern. There's a multiple, let's say, cluster 9, where the arginine stands -3 position. That's a very common pattern as well. So anyway, but the question which kind of people asked a lot, it's a bit of why specifically this serine threonine was modified. Why specifically this serine threonine was chosen by kinases to be phosphorylated? And of course, very kind of direct answer would be because of the sequence context. But how informative the sequence context? Having proline, is it actually very informative? Is it, if having proline guarantees that you're going to be phosphorylated? You see, all these contexts are not very super informative. And my task, I try to actually thought about structural context, not just sequence context, but I try to find out some, maybe some gloves, caveats of the proteins which being detected with kinases. Maybe kinases do care about sequence, but maybe we care more about 3D structure surrounding this serine threonine. And I failed miserably. This was a super horrible experience. And slowly kind of, we found out why. And of course, there's some papers which talked about that. You're just stupid enough to didn't find this. But what basically we found out, that's if you make, again, so if you take basically any structural predictions, I think it was AlphaFold in that case. So you probably know it's like AlphaFold for each amino acids highlight how sure about structure of this amino acids the model is sure about. So from very high, from 90 plus to the high, low, and very low. And now if you plot all amino acids distribution will look like the black line. So indeed, many of them have a low structureness, but most of them indeed have a high structures. But now if you plot all STYs, the distribution, the gray distribution will look exactly very similar to the all amino acids, right? It's very close to that. But now if you plot all STYs, which we found being phosphorylated, or even phosphorylated in at least one tissue, that's the like light blue, but in all tissues means it's very ultimately present in every tissue of the mice which we studied. So you'll see that all these phosphorylation sites will love to be in non-structured regions. And now you ask me a very good question. Where's the chicken? Where's the egg? Right? So is this phosphorylated because it will change structure and fold in something meaningful? Or it's being modified because it's so flexible, so floppy, and that's why kinase just has a higher chance to find this floppy part of sequence? Super cool question. I have to say, I don't know. I found even super ridiculous papers of 2006 when people tried bioinformatically predict which serine threonines will be phosphorylated. It sounds like a very stupid task because it sounds like tissue-specific information. But what we were doing for the overall brutal human body. And you know, the best predictive thing was structureness, prediction of intrinsically disordered regions. Apparently, intrinsically disordered regions love to be phosphorylated. But again, chicken egg, I don't know what's the relations there. I think it's a very interesting challenge. But to make some kind of PTM-aware structure prediction or PTM-aware modeling, I think it's super cool. Somebody should do that. Okay, and the last few thoughts. It's a bit of philosophy, but I kind of, when I talk with my students, please try first with basic statistical models and slowly accelerate complexity. Yeah, unfortunately, people start from another end frequently. And that's end up with ridiculous cases when people showing that some linear regressions outperform machine learning, and you know, and no GPU and gigawatts needed. So most of the users do not have advanced GPU computer power, and I must say I do struggle with this because, like, if I do develop software which people use to analyze the data, and to rely on the fact that people will have GPU, it's actually a bit too fast stretch. People can use clouds. It costs money, and I don't know, but in the lab you would like to try different things and don't think about money. That's mindset, right? And it's a bit hard. A few thoughts. I honestly happy that in our field, I would say people are really don't treat machine learning as a black box. We're constantly trying to interpret that. We're not, the papers are not end up with comparison of multiple machine learning models and some quality metrics. Indeed, people trying to validate, interpret, and I hope reviewers also helping to maintain this standards in the field. Yeah, interpretation first. And also, I a bit paranoid, and that's probably everyone have to be paranoid, data leakages happening everywhere. So just keep in mind this, just try to think smart how to design test train split. That's basically half of the problem, the model which you're trying to build. That's it. So I didn't make acknowledgements slide, but many people have been involved here. I didn't highlight many papers. I just highlighted what I saw or reviewed. So it's something which I was familiar with. And this is very personalized thinking, of course. It's not scientific review. Yep.
Yeah. Thank you so much for the talk. Yeah, that's actually what it's supposed to be. So the initial idea behind the meetings that we have is to have to share the personal opinion. So thank you very much for your perspective. That's actually what we want to hear here. And maybe if you don't mind, I will ask again the standard question that I ask everyone. What do you think about the place for the large language models? Because I saw, yeah, and I heard that you mentioned the protein language model, obviously, and you've mentioned the nice idea of applying transformer architectures. Which, for those of you who are less familiar with this architectural stuff, transformers is essentially the core of GPT and all of the sort of fancy LLMs. And here, the same idea being applied to this de novo sequencing of peptides. That's very, very nice. But do you see a place for GPT and other models like that somewhere in the proteomics pipeline? For example, in interpretation of results or something? Do you have any opinion as to whether there is a place for these kinds of tools?
You also noticed I separated on two parts: machine learning for method development and machine learning for application, right? And indeed, it's very different machine learning. I'm sorry. From one perspective, amino acid sequence, there's some interlink relations between even long-standing amino acids. And basically, if LLM is able to find or modulate these relations between words, why we cannot do this for amino acids? It's very meaningful usage, to be honest. But again, I would not scuba dive immediately to deep learning or LLM to start with. I would say maybe right now the protein language models actually helping a lot to extract meaningful features, meaningful in terms of kind of how it was trained in the first place. But anyway, so it's trying to help to extract some meaningful features which can be further used for your useful tasks. So fine-tuned for your useful task. Yeah, that's kind of, I feel like, helpful. It's already being used and used a lot for interpretations. And again, but what's your downstream task? Your downstream task can be super crazy. So imagine if you see differentially expressed proteins or proteins which being modified in your data sets, and you ask yourself, what's the common feature of all these proteins? Previously, people use Go enrichment or GCE enrichment, yes, but maybe we can at certain moments try to use just, protein language model assisted binary classifiers. I think that's a real thing. It's and actually it would be interesting to see. Yeah, so I would be all time skeptical. Try modest first, and if there's a need and if you see it's useful, yeah, try LLM. But yeah, I still believe I would not ask, to be honest, LLM to do a t-test for me. I would rather do it myself in Python or R. Yep.
Thank you. Yeah, there was also a question from the chat. So I think the first part of the question is whether you have tried to use machine learning to predict or detect tumor-specific neoantigens. So this is the first question.
When you're saying antigens, do you mean, sorry, HLA peptides, new HLA peptides? I know it takes some time to answer. So if it is, then I'm just writing grant on that. So I can talk a lot about that, but please don't make my life painful. So yeah, that's a lot of things being done there, and it's especially HLA peptides. Sorry, we're not tryptic peptides. We don't have arginine lysine at the end. We're super short. We're 9 amino acids. Machine learning is just super fruitful soil to use there. So it's perfect application, to be honest, because it then can predict intensities, and predict intensities just gives better chances to separate better target versus decoy, means finding better peptides and being more sure about quality of these identifications. Yeah, that's definitely used a lot. Another thing, how to integrate PTMs, post-translation modifications. That's an open question. People struggling. Let's see, maybe some graph-inspired representation of PTMs. Maybe. Let's see.
And the second question, again, in that same post in the chat, is a kind of structure of a specific part of a protein, how visible it is in the mass spectrometry experiment, because it can actually be an additional bias that affects the PTM-related studies.
Yeah, so how PTM, so yeah, predicting digestibility, solubility, flyability in the context of PTMs, that's the next step. That's indeed, it's very much related to one super interesting question. It's about occupancy. So imagine you have some site which you know being modified, and you have 100% of molecules. Out of this 100% of molecules, how many molecules have this site being modified? How many molecules actually present with this site being modified actually? And that's called occupancy, phosphorylation occupancy, glycosylation occupancy. Again, unfortunately, you cannot compare a modified peptide to peptide just one-to-one, just like that. It doesn't work so easy. Unfortunately, all these biochemical properties which being changed thanks to the PTMs were affecting a lot how much we detect, like affecting intensity in a very non-linear way. And that makes whole comparisons really complicated. Especially the question is how PTM affecting protease specificity, how PTM affect solubility, how PTM affect detectability, like flyability. So that's all these very much open questions. There's some efforts, but again, I think it's by the field, it's not acceptable anymore just to make black box and just sell it. No, enough. So you have to kind of validate the results. You have to interpret your results, and that's, I think, like makes the whole field interesting. Thanks to that. Yeah.
Okay. Thank you so much. Yeah, I think that there are no more questions in the chat, and we are also out of time for today's meeting. So thanks to everyone who have joined us today. Thanks to Pavel and to Yana again for the very nice talks. It was a pleasure to hear your perspectives on the topic. Yeah, thank you very much. And yeah, see everyone next time. Next time we have the concluding meeting of this session.